Okay, it's okay. Okay, Assalamualaikum and very good afternoon to everyone. Thank you for being with us today, and I hope everyone is well and safe at this moment. So, as Prof. Jenny said, I'm also the coordinator for this seminar from now on. So if you have any anyone that you want to invite for the seminar, then you can email me at my email here, fadlihamdan@um.edu.my. So for today, I'm going to be the presenter, and the title is Production of Novel Alleles Using Directed Homologous Recombination Technique. So for today, I would like to touch on three main points. The first one is why is genetic diversity is important. Secondly is on the use of homologous recombination application in plastid transformation or chloroplast transformation. And finally, looking at the possibility of exploiting homologous recombination or HR to produce novel alleles or new variants, basically, and how can that be done? So, genetic diversity is the variation in the genetic composition among individuals of a population, species, or community. And this is true in humans, plants, and other organisms as well. And within these variations, there are hidden characteristics that are as a result of all the biological information from thousands of ancestors before us. And so in the context of agriculture in plants, genetic diversity is important because it helps to increase the survivability of plant species by allowing possible emergence of new alleles that might not be, emerge and might have benefits to us. So it could either be, for example, increase in yield or higher resilience towards diseases, pests, and other stresses. Okay. So in agriculture, why this is important? Because as I said before, it helps to increase the chance of survivability, and in terms of our need, we want to have crops that are improved in hopefully in yield or other environmental resistance. So I just want to share this slide because I think it's quite interesting. Maybe some of you or all of you have seen this before. This is an ancestral plant, a very old plant, the fruit part of the plant actually, and it has a few kernels, and it tastes like dry raw potato. It has a relatively low sugar content, about 2% and 20% starch, and currently there are 8 known varieties. So maybe you can guess which what plant is this. It is so what happened is that through selective breeding for thousands of years, in particular more than 9,000 of years of carefully selectively breeding these plants, farmers have managed to sort of produce a new version of these plants, crops that are very much suitable for our consumption. And there are the difference between the modern version of between the modern version and this ancestral plant is that it's on roughly about 5 different genes. So the modern so this plant is called Teosinte. I'm sure many of you knows about this. And the modern version of it is the modern corn. As you can see, modern corn almost looks like nothing like Teosinte. It is easier to peel, has a sweet and juicy taste, more sugar content, about 6%. And also at the moment there are about more than 200 varieties in the world. So this is through selective breeding technique, in which the genetic content of the plants have been altered through these conventional techniques. to create crops that are more suitable for our consumption. So this, when we talk about natural corn, someone might actually refer to this natural corn, teosinte, and the one that we have is actually artificial corn in the sense that artificial meaning that there's a human intervention in creating this crop. It wouldn't appear or it will take such a long time to appear by spontaneous mutations or breeding. Mix of parental genes to create these crops. So in the nature, it wouldn't appear without human intervention. So just a little bit more on teosinte. It's native to Mexico, Guatemala, Honduras, and Nicaragua. And the domestication of this crop, what happened is that these plants were taken to the southern part of America before being hugely domesticated around the world. And as we know now, it's one of the most cultivable crops in the world. The modern corn. So this is just another picture showing the evolution of teosinte becoming the modern corn that we have today. And there are so many different varieties in the world right now with different of corns with different taste, color, texture, and so on. So as you see the corns in Pasar Malam, so we have to be thankful of the works of so many farmers and scientists for thousands of years to get to this the sort of best version of corn that we have, which is very tasty and juicy instead of just dry raw taste of potato. So this is another example, but I won't talk too much on it. But so as you can see, this is the modern watermelon, artificial watermelon, and then compared to its ancestor, the natural watermelon, which is very small in size and extremely bitter taste compared to the ones that we all love, the modern watermelon. And different varieties as well for watermelons, slightly different shapes and colors. So in summary, the use of selective breeding for thousands of years have been done to, although indirectly, not knowingly, to alter the genetic content of crops for food and feed consumptions. We have been doing this for so many years without the knowledge of, even before the knowledge of heredity and genetics and so on. One main feature of this conventional technique is does not involve insertion of foreign genetic materials, unlike the modern technology. Also, we do this because we want to change the texture, size, color, and taste, and possibly yield to make it more, high yielding than the previous ones. And this process can take a very long time, as you can see in the case of corn. It took thousands of years of trial and error to get the most desirable traits. So this is an effort of farmers and scientists to increase the genetic content, the diversity of plants or crops so that the traits with the desirable ones would be able to emerge. Okay, so let's look at the second part of my presentation, which is on homologous recombination. So in short, homologous recombination, or HR, is basically involves the exchange of genetic material between two highly similar or identical DNA sequences. So it is very important because this, one could argue that this is possibly the one that makes us all unique, because each of us unique, because it's the exchange of genetic materials are the ones that makes that creates the variation in the offsprings, and also to increase the chance of survival in the changing environment. So as you can see in the figure here, briefly, you can see the blue region, small region, on the red sister chromatids change from the original one, the blue sister chromatids. So these two are homologous chromosomes, and so the crossover or the recombination site is between the two small blue region and the red small region there. So I have reason why I show this, because this technique has been used in plastid or chloroplast transformation to edit specific genes. So the basic design is that we have a DNA vector containing the gene of interest, usually fused with the marker gene. And then what we do is that we have this left arm and right arm flanking this DNA cassette. And then what happened is that when this DNA vector is integrated into the host plant, the left arm and right arm will recombine, cross over with their similar sites on the chloroplast genome, and as a result, integrated the DNA cassette into the genome. So this has been efficiently used in chloroplast transformation. But what we also found out that recombination, when we put DNA repeats inside the DNA cassette, right? And these DNA repeats have either identical or highly similar sequences, as shown in number 1 here. So if you have the DNA cassette integrated into the host genome, so let's say this rubber band looking thing is the plastome or plastid genome. So let's say it has the marker gene in the middle, flanked by DNA repeats, and these repeats are highly similar in terms of its sequence. So what happened is the two DNA repeats will start to recombine. There's a crossover event happening between these two repeats, and then finally we'll have the marker gene excise removed from the plastid genome. And what's interesting is that the DNA repeats, once they recombine, will have the new novel gene inside the plastid genome, as shown in number 4 here. As you can see, theoretically here, if the crossover happen in the middle, so we will have 50% from each of the original direct repeats. So this is a new gene. I'll explain a bit more on that. So yeah, so as I said, depending on the crossover events, right? If you might have single or double crossover events. So for double crossover events, what you'll also get is a mosaic type of DNA integration. Mosaic meaning that DNA patches of DNA integration into the new gene. It's probably the term is probably not correct. It's not new. It's the same gene, but has multiple single nucleotide polymorphisms or mutation sites inside the gene now in the plastid genome. So the next question we ask is that how can we exploit this? Can we exploit this homologous recombination pathway, native pathway, to not only remove the marker gene from the plastid genome, but can we use this to produce novel alleles, a new variant of the same gene? So what I did before was that I tested the RBCL genes, which encodes the large subunit of an important enzyme in plants, rubisco enzyme, which is involved in the photosynthesis process. So what I did was that I designed 2 RBCL genes, so indicated by these green bars here. And as you can see on RBCL-A, there are multiple yellow lines indicating the polymorphism sites or the mutation sites that are present, that are pre-inserted into the gene first. So once this construct is inserted into the chloroplast genome, depending on the crossover sites between these two repeats, you will get different versions of the genes, new genes, novel alleles. So let's see now as RBCLC here, you get these yellow lines at the 5' end of the RBCL gene, and then the rest coming from RBCLB, because the crossover event happened in the middle. Yeah, roughly in the middle. For RBCLB, you see 3 mutation sites near the 5' end, because the crossover event took place at nearer the 5' end of the RBCL repeats. So these two are novel variants, wouldn't exist in the wild without this technology. So currently, what people have been doing is that in order to study gene expression and to test these mutation sites, for each mutation site or each pattern, they need to test separately, which means for each gene variant, they need to construct one DNA design, DNA transformation vector. And this could take a long time to make and quite laborious work involved. But with the proposed technique, what happened is that we just need one construct, and then depending on the and then in one of the DNA repeats we put this DNA mutation sites, and then depending on the crossover event between the two DNA repeats, we'll get theoretically multiple gene variants of the same gene, but different pattern of integration of the mutation sites. So this sort of speed up the whole process of studying the gene expression. Also, this application, this tool has been applied in Rubisco, as I mentioned earlier. Rubisco is a very well-studied protein enzyme because it is involved in photosynthesis, but the turnover rate, its efficiency is basically very, very low, because there's another process, another competing process, also initiated by Rubisco enzyme, the photorespiration. So many groups have been studying Rubisco by introducing a number of mutation sites and to see whether they can try to improve the efficiency of Rubisco. But as I said before, it will take for each DNA mutation, they need to design a unique construct for that. So someone might ask, how do we decide the mutation sites that we want to test, right? So one of the ways is to look at natural variations in other plants that have Rubisco with higher efficiency, and try to introduce those mutations, those differences into the host genome that we are looking at. And this is just a 3D model of the large subunit of Rubisco enzyme with all the DNA alteration sites that were introduced. And so the steps involved in doing the transformation. The first one is to do to insert the DNA construct into the host plant, and then secondly we'll do a first round of selection with antibiotic, and then next we'll try to isolate the resistant cells with green callus, which means that the constructs are present in the transformed cells, showing that they have the antibiotic resistant marker genes with the DNA repeats. And then in step 4, we do a second round of selection with stronger antibiotics, basically. Next, we remove these antibiotics from the media to let the plants to root, and before transferring it to soil to be analyzed. But I want to point out that there's another step here, in between step 4 and 5 here, which is very important, because this is where when we remove the antibiotic, right? What happened, as I said before, it allows for the DNA repeats to start aligning and recombine. So depending on the crossover size, you get these types, different types of novel or new variants. You might get 3, 5, 10, 20 or more, depending on the crossover events. So previously, when I did this, I managed to isolate eight new RBCL variants, alleles, with different pattern of polymorphisms. So there were 19 polymorphisms, 19 sites here. As you can see, the lines, the black lines representing the mutation sites, they are present all over the RBCL, the novel RBCL alleles. So there are 8 different variants, and these were isolated from 21 different transformants with the mutation sites introduced. So they managed to recombine, and showing this pattern of integration. So basically, out of 21, 8 of them were unique alleles. And based on my calculation before, if you have 19 sites, theoretically, on paper, you would get, you might get up to around 500,000 different variants that you might be able to might be produced. But of course, there's so many different factors to be considered in terms of the length of the DNA, the homology between the two DNA repeats, the length of the repeats itself, and so many other things. So you probably wouldn't get until 500,000 different variants. That's too much. But for showing that this can be done, proof of concept, it is quite possible, because there were 8 different variants produced. If you look at the previous slide, so that if using the current technique of site-directed mutagenesis, you need to design 9 different constructs to test these 9 mutation, 9 patterns of mutation sites that you want to look at. But by using this proposed technique, this was done in a single transformation event with 1 DNA construct. And on top of that, the antibiotic resistant marker gene was removed from the final transformants. Okay, so as we all know, the advantages of doing transformation on chloroplast genome, using this technique, it allows us to find or to alter genes that might improve grain yield. Also, we could use chloroplast genome-sized bioreactors to produce vaccine antigens or therapeutic proteins. And vaccine, if I'm not mistaken, this has been done recently, plant-derived COVID-19 antigens using chloroplast transformation, and to exploit plastids also to produce plants that might be able to clean up toxic contaminants from the environment, or also known as phytoremediation. And finally, to use these transformants as biofactories to produce beneficial products such as biomaterials, enzymes, and so on. And so finally, so in summary, genetic diversity is good because it increases our chance of surviving in surviving in the environment. It's true for plants as well. So plants that will be able to be resist to different kinds of abiotic and biotic stresses. Secondly, the conventional methods now, such as selective breeding, and not just selective breeding, there are many more conventional techniques, you know, like seed saving, plant hybridization, mutational breeding or variation breeding. All these, the purpose is to increase the genetic pool of crops to get to try to create or produce crops that have these desirable traits. And finally, modern technology, such as using this HR directed method, it also increases the pool of the genetic pool, but to using a more specific DNA alteration method. And hopefully it will be relatively faster and also a bit less risk of integration of unwanted mutations. Yeah, I think that's all. I think that's all for me. Thank you.
Thank you, Dr. Fadli. My apologies to you because I have to run off to another meeting at 3. So I've asked Dr. Tan Boon Chin to take over for the Q&A. Very nice work, and I think it's because I used to do yeast work, but we do a lot of homologous recombination. So it's really nice to see the homologous recombination story again. Thank you very much.
Welcome. Thank you.
I hand it over to Dr. Tan now. Thank you.
Okay.
Okay, thank you very much for Dr. Fadli for sharing her his research with us. So I now open Q&A sessions. If anyone who have any questions, you can just unmute your mic and then just ask the questions, or if not, you also can type it in the chat box. So perhaps I can start with questions, general questions. So so for this HR, right, is that really difficult or challenging of you know, using this HR to do the direct mutagenesis compared to the conventional one?
Well, it depends on I think the intention of the research. So let's say if you want to use the conventional one is one of the disadvantages is the possibility of off-target introduction of mutation sites, for example. Depending on this HR technique, it's very specific because it has these flanking arms targeting right at the site that we are only interested in. With the conventional methods, you try to introduce these mutations without knowing if there's any other effects on other mutations as well. So, but as I said, depends on what we want. If using conventional methods, you just blast these agents, I mean transforming agents, and see whether you could and screen out as many as you can, right? So if you're not worried about those kind of target sites, then maybe it's easier that way than using HR. But HR is more specific and less risk of unwanted mutation integration.
Thank you. Is there any other questions from others?
Yeah, I have one. Fikri here.
Yes.
Thank you, Fadli, for your presentation. So I just wonder, I don't know whether, I just wonder if you can scale this one up by using multiple construct to target multiple genes, but still using HR as the basis.
Yes, I actually discussed this with my previous supervisor. But for now, the thing is, if you do that, then you have to do in consequently. So you
you can't just like put 5 construct and then let it go wild and see whatever variance that you get. Okay.
Yeah, so it will take more time as well.
Yeah, because if
But of
it's
course
a
you can do,
if it's a multigenic type of trait, and you have to you need to have to be able to target those multiple genes.
Unlike the recent innovation of CRISPR technique, I think you can target multiple genes now with CRISPR. But the intention, the goal of this is quite different. It's focusing on just one gene, but allowing for different mix of mutation sites, so that we speed up the process of selection later. So from what I understand, for number of genes, if you want to look at, then you have to do consequently, so one by one.
All right, thank you.
Thank you.
Thank you, Dr. Fikri. And any other questions from others? No, I don't see any in the chat box also.
Yep, I don't see also.
Okay. So, right, if no further questions, so we thanks Dr. Fatri for his interesting presentations and willing to share with us his research. And just a reminder to all, so please, if you go through the chat box, there's a link for you to fill up the evaluation form for today's seminar. Okay. Right. So thank you, Fadri, again, and thank you everyone for attending the SEBA Expert Seminar today. So we hope to see you again in our next seminar. So with that, thank you very much.
Thank you.
Thank you very much, everyone.
Thank you, everyone.
Thank you all.